So hopefully everyone's had a little bit of break. You're feeling a little refreshed and ready for the second session of this conference, where we're going to be started off by Professor Martin Collinson. He's going to tell us about some of his great research he's doing at the University of Aberdeen. So I'm going to hand over to you, Martin.
Hello, everybody. Thank you very much. Thank you very much, obviously, for asking me to present today. What I'd like to be able to do for you is to describe some of the work that's happening in my lab, where we're looking to develop therapies for aniridia-associated keratopathy. Slide here. This slide has a picture of the eye of somebody with aniridia and their experience, and if you can see, quite a severe corneal opacity. And this is what we call aniridia-associated keratopathy, also called aniridic keratopathy or aniridia-related keratopathy. That's all the same thing. And as you're probably aware, this corneal opacity is a progressive condition that can happen in most people with aniridia to some extent as they get older. The surface of the eye starts to vascularize, it gets blood vessels and can become inflamed, and this can lead to loss of vision. Not everybody will get the keratopathy as bad as this person here, but of course, and as we've heard earlier today, the lack of certainty about the extent to which keratopathy can affect people's vision in the future is a very significant source of anxiety both to people with aniridia and to their parents. And what we're trying to do in our lab is to be able to prevent this from happening, or in people where it has already happened, potentially to reverse this opacity, to maintain vision long into adult life. Now, for those of you who are familiar with aniridic keratopathy, you may be aware that it is treated as a so-called limbal stem cell deficiency. This slide has a picture of two eyes. Top one is a healthy, non-aniridic eye with a nice blue iris. And the lower eye is a vascularized, inflamed, essentially opaque cornea due to a known limbal stem cell deficiency. And to explain a bit of the background to this, the front surface of the eye, the cornea, is essentially a specialized skin. And just like for the rest of the skin, cells fall off all the time, just to normal wear and tear due to abrasion, and they have to be replaced. And our skin has stem cells that divide to replace the cells that are lost, and so does the cornea. And you may be aware that the stem cells that maintain the cornea live around the edge. The boundary between the blue bit on this side and the whites of the eyes is a population of stem cells that are dividing all the time, fairly slowly, to replace the cells that have been lost. And in people where these limbal stem cells don't work, either because of disease or injury, alkali burns, for example, the surface of the cornea can't be maintained. Cells are falling off, but they're not being replaced properly. And you get the sort of opacity and vascularization that you see in the bottom eye here. And this looks very much like aniridia-associated keratopathy. And historically, the condition has been treated as a limbal stem cell deficiency, and many people with aniridia end up getting limbal stem cell transplants from non-aniridic individuals, and these can help with the maintenance of vision for a few months or even years, but they're not a permanent solution. In order to study aniridia, we've been using in our lab a strain of mice that has PAX6 mutations like people with aniridia do. Not everybody is totally comfortable with the idea of using these mice, but these are perfectly happy, healthy mice. But just like people with PAX6 mutation, they do have all sorts of changes to their eye. So there's two mice on this screen. The one on the left is a normal, healthy mouse with big bright eyes. And the one on the right is a mouse with aniridia. It's got a PAX6 mutation. You may notice its eyes are smaller. It's the so-called small eye mouse. We're not bothered about the size of the eyes. What we do like about these mice is that they have aniridia, they have lens cataracts, and they have a kind of progressive keratopathy. The cornea, the front surface of the eye, goes cloudy over time as they get older. And it really very faithfully recapitulates the human aniridia associated keratopathy symptoms. So I've had PhD students and other people using these PAX6 mutant mice we've spent pretty much 20 years working out what the link is between PAX6 mutation and aniridic keratopathy. And we've had students asking the questions, is this really a limbal stem cell deficiency? And as with most things in science, the answer is maybe, but maybe not. And here's a little cartoon of some experiments that a PhD student of mine did. Some little cartoon mice. The mice in the top row are normal wild-type mice, and the mice in the bottom row are small eye mice with a PAX6 mutation. And what one of my students did we put the mice under general anesthetic, and she gave them a little scratch to the surface of the eye. The idea is that limbal stem cells are meant to respond to eye wounding by making more cells to replace those that are lost. And in a normal wild-type mouse, that's what happens. If you scratch the surface of the eye, within 24 hours, you'll see this mouse has got one yellow eye, which we've used to represent activation of the limbal stem cells. So we can see the limbal stem cells dividing at high rate in response to a corneal wounding. And after 48 hours, the limbal stem cells in both eyes have activated, and they're really dividing as fast as they can to repair the injury. We've already seen that PAX6 mutant mice do have limbal stem cells, and they do divide. And when my PhD student was able to wound the eyes of these mice, again, just give them a tiny little scratch to the cornea under anesthetic, the limbal stem cells, they do wake up after 48 hours, that they respond to the injury and they're dividing to produce more corneal cells. But the difference was that they took longer to do it. After 24 hours, there was no response for the limbal stem cells. So there is a deficiency here. It's pretty mild. People and mice with aniridia do have limbal stem cells. Those stem cells can divide, but they take a little bit longer to respond to injury. And the question is, can that explain the whole problem with the cornea going opaque in aniridia? And we believe the answer is no. Again, this slide again just shows another person with aniridia-associated keratopathy, with blood vessels growing into the cornea and a big opacity in the cornea. And that the reason why this is happening is not because these people don't have limbal stem cells. There's much more going on. There's a whole lot of things going wrong in the corneal epithelium itself. And this is what we spent 20 years trying to work out what the problem was. And rather than give you a summary of 20 years of research, I'll just describe briefly what the conclusions were. And the problem essentially is that the PAX6 aniridic cornea, the surface is fragile. The cells don't hold together as well as in normal non-PAX6 deficient corneas. And what that means is that the cornea gets small lesions, small holes in it. This is a figure that I lifted from an old paper, and it's not too important to, but what four of the panels here are showing are scanning electron microscopy. It's a really clever imaging of the surface of the cornea. And the first two, the top two panels, are really boring. It just looks like it just looks like nothing really, because these are normal, healthy, wild-type corneas, and they're lovely and smooth, and there's nothing to see. But the lower two panels, C and D, show corneas with holes in them, and these are from the aniridic mice. So cells are being lost too fast because the cornea is fragile and it develops holes in it. And because of that, it doesn't really form an effective barrier to the outside world. I don't know if any of you at the ophthalmologists have had, they use fluorescein, sort of fluorescent green dye, to determine whether the surface of the eye is healthy or not. Because a normal healthy cornea won't take up fluorescein dye, whereas one where the epithelium is lesioned, it will. And I don't know if you can see down here, this is a section for a mouse cornea. All the cells are blue, and there's a little stripe of green at the top where we've put fluorescein on the cornea, but it hasn't penetrated. Whereas this is a normal healthy mouse, and this is a PAX6 deficient mouse, and you can see that the yellow dye has spread throughout the whole cornea, that it's no longer forming a barrier. So this is essentially the problem with aniridic corneas. They don't form a proper barrier to the outside world because they're fragile. So stuff gets in, and it's not normally fluorescein, but it can be oxygen, noxious agents, dust, viruses, fungi, all sorts of things that are bad for the cornea that the cornea normally keeps out, but that the aniridic cornea is just slightly less efficient at doing. We came up with what we call our triple whammy model of aniridia pathology. And it's summarized in this diagram on the right here. At the top in blue is the start of the process, a PAX6 deficiency. So people with a mutation in one of their PAX6 genes, that means they don't have as much PAX6 as normal. And down at the bottom is the outcome, corneal opacity. The cornea is no longer fully transparent and people start to lose vision. And in between are the things that we think goes wrong. And it's summarized on the left in the yellow writing with the so-called triple whammy model. And whammy 1, as I already explained, is that the corneal epithelium in aniridia is fragile, and it loses its ability to act as a barrier to the outside world. The second, so it's under stress. Oxygen gets in, pathogens get in, and the cornea is under stress. The second whammy, which I haven't mentioned yet, is that normally when tissues are under stress, they produce protective enzymes that stop that stress from damaging the tissue. And for a number of reasons, aniridic corneas have lower levels of these stress-responsive enzymes. So they are more stressed, they're suffering more stress, and they produce less of the enzymes that respond to that stress. So that's what these two yellow boxes are showing here. We have a fragile corneal epithelium with less of the protective enzymes that it needs to, and this leads to increased stress. Proteins get oxidized that shouldn't be oxidized. All sorts of cell signaling happens that shouldn't be happening. And this leads to a kind of chronic wounding state, even in the so-called healthy aniridic cornea. It's constantly developing holes and then healing those holes and then getting more holes and healing the holes again. It's constantly in a wound healing kind of state. And this leads to two things. one of which isn't obvious. That it effectively reduces the amount of PAX6 that's available. Because PAX6, as you might know, is a gene. It's a protein that controls the expression of a whole pile of other genes. So it's a DNA binding protein, and DNA is in the center of the cell in the nucleus. But what stress does, and one thing that we showed, is that it drives the PAX6 out of the nucleus. So even the PAX6 that's there in the cells can't do its job properly because it doesn't get access to DNA. So that's what these arrows pointing backwards are showing. It exacerbates the PAX6 deficiency. And because of the constant chronic wound healing state, eventually the cornea becomes vascularized, blood vessels grow in that shouldn't be there, it gets inflamed, water gets in, it disrupts the structure of the cornea, and over time you get a corneal opacity. So this is 20 years of work that we have spent trying to work out why a deficiency of PAX6 leads to corneal opacity at all. The question is, that's very nice to know, but what do we actually do about it? And our vision is that when a person has aniridia, we want to be able to give them therapies, eye drops or something similar, that will prevent this triple whammy model from taking effect and will break the link between PAX6 deficiency and corneal opacity, such that good vision can be retained into late into adult life and basically for the rest of your lives. And we've taken two strategies by which we're attempting to do that. One of which, the left-hand side here, is direct antioxidant therapy. So if the problem with the cornea is oxidation and stress, then there are chemicals we could use to reduce that stress. And the other kind of obvious strategy, the second one here in yellow, is the restoration of PAX6 activity. So that people with aniridia are deficient in the amount of PAX6 you produce. So can we restore that back up to normal levels? And what the next slide is going to show is 3 streams of research that we have been undertaking to try and address these problems. And the number one on the left here is talking about two drugs that are in the public domain. Has been published as increasing PAX6 dosage in the eye. One's called duloxetine and one's called ritanserin. And they've been shown in tissue culture, in the lab, in cultures of eye cells, that they can increase the amount of PAX6 that is produced by cells. And I'm going to describe some work that we've done putting these drugs into aniridic mice to show what they can do. The second stream in the middle here is something I'm not talking about too much about today. But those of you who've been involved with the aniridia therapy scene may have heard of a drug called ataluren, which it's a type of what we call a read-through drug, which allows cells to ignore mutations in PAX6. So even if you have one copy of the gene is mutated, in the presence of so-called read-through drugs, the cells will ignore that mutation and produce normal PAX6 anyway. And there is a drug called ataluren that it's one of these read-through drugs, and it's been trialed for aniridia. It didn't really do a lot of good. Nevertheless, it's not the only read-through drug out there, and we are testing a whole pile of other read-through drugs to see if they can help people with aniridia. And the third stream on the right here is the development of antioxidant therapies that will allow aniridic corneas to just get out of this vicious cycle of perpetual wound healing, to be able to sort of calm down and allow the regenerative capacity of the cornea to be restored, such that the loss of opacity doesn't occur. And what's crucial about these three research streams is that we're not coming up with new drugs. What we're doing is finding approved drugs that are already being used to treat people for other conditions. Like duloxetine, for example, on the left here is a known safe antidepressant. And we're saying we're taking these drugs that are already, we know they're safe to give to people. We know that they've passed clinical trials for other things, but we're asking, can they help people with aniridia? And by taking this strategy, what we're hoping to be able to do is to accelerate the process by which we can identifying a drug that helps with aniridia can be taken through clinical trial and given to people in the minimum amount of time. Because as you're aware, with vision loss in aniridia, time is critical, and there are thousands of people out there that could benefit from a therapy whose eyesight is frankly not getting any better. We want to minimize the amount of time that it takes to get drugs to helping people with aniridia. So I'm just going to do a bit of science. I'm going to describe an experiment that we did last year, actually, where we gave normal mice. So here's our two friendly mice again. The one on the left is a normal wild-type mouse, and the other is one with a PAX6 deficiency. And we gave them the drugs, these two drugs, duloxetine and ritanserin, together in their drinking water for 4 weeks. And we monitored their eyes during the process. We actually monitored the health of the mice to make sure that it wasn't causing them any harm or side effects. Like I said, duloxetine is an antidepressant. Ritanserin is an antipsychotic. These are quite powerful brain drugs, but the dose was very low. And we put these drugs in the mouse's drinking water and saw, well, did their eyes get better? And the long story short is that yes, they did. On this slide here, we have some mouse eyes. Mouse eyes have all the same bits as human eyes, but they're not nearly as pretty, really. And they have black irises, and they're much more like footballs, because whereas we are looking at things that are very far away, mice are basically looking at sunflower seeds that are very close to them, so they have a different shape eye. Otherwise, that their eyes are like ours. And at the top of the screen here, there's two wild-type mouse eyes, lovely and shiny, lovely and clear, a nice big black iris. And bottom left here is from one of the aniridic mice, and it still has an eye, obviously, but there's severe loss of the iris, so iris hyperplasia or aniridia, and the surface looks a bit rough. And they've had fluorescein treatment, so it's yellow because this fluorescein has penetrated the eye in a way that it didn't with the wild types. And you can see there's a bit of a scar in the lens, maybe. And this is the heterozygous mice that didn't get the drugs. These just got the controls. And this is a normal aniridic mouse eye with lots of problems. And this is an aniridic mouse eye that got the ritanserin and duloxetine combination therapy. And I hope you can see it's better. The iris doesn't grow back. I mean, that's gone. There's nothing we can do about that. But the surface of the eye looks nice and smooth. The barrier function has been restored, so it doesn't take up fluorescein very much either. And essentially, this mouse with the duloxetine ritanserin therapy has better vision than this mouse that didn't have it. Here's some science bits. Science bit 1. These are tissue sections of the mouse's corneas, and we've labeled the PAX6 protein fluorescent green so we can see it. And top left is a wild-type cornea, and you can see there's a streak of green cells that has the normal levels of PAX6. And this down here, bottom left, is a heterozygous, so PAX6 deficient cornea. And if nothing else, you can see that there isn't as much green in these cells as there is above, because these guys don't have as much PAX6. But going to bottom right, the heterozygous mice after treatment, they have much more PAX6 in their corneas. So the drugs seem to be working. They're increasing the amount of PAX6 that the cells can produce. Another science bit, there's two things on this graph, one of which is a tissue section through the cornea, and you may be able to see that in the epithelium here, there are some bright pink blobby cells. They shouldn't be there. These are conjunctival cells. So one of the problems with aniridic corneas is that the epithelium of the whites of the eyes, the conjunctiva, starts to encroach onto the corneal surface, and it's not transparent, and it's vascularized, and that's bad. So what we want to do is to prevent this conjunctivalization from happening. And this is what this histogram on the right here is showing. Basically, the bigger the bar, the worse it is. And we have control mice with no conjunctival cells, with or without drug. And this is an aniridic eye from the mice, and they all have conjunctival cells. But after drug treatment, it's been reduced. Something less than half of them now have conjunctival cells. So within 4 weeks, we've managed to push the conjunctivalization of the aniridic corneas backwards just by giving the mice these drugs in their drinking water. And that's lovely and everything. But I've already said that these drugs are powerful. Duloxetine is a powerful antidepressant. Ritanserin is a powerful antipsychotic. Our vision, as it is that when a baby or a young child is born with aniridia, we can start giving them drugs that will prevent the deterioration of vision. But we can't be giving babies and young kids powerful antidepressants just for the ocular, the effects on their eyes. What we want to be able to do is to develop eye drops, so that kids aren't getting a systemic dose of these powerful drugs. They're getting just a very small topical dose delivered right to where it's needed, to the eye. We're not as far on with these experiments, but we have been, we are doing a series of experiments where we give the mice eye drops. So mice with aniridia, PAX6 deficient, twice a day, putting a little blob of gel in their eyes that contains either nothing, the controls, or that contains duloxetine and or ritanserin. And this is an experiment where we did it for 2 weeks. Again, aniridic mouse eye on the left, looking a bit rough. This is a control. Aniridic mouse eye on the right that's had duloxetine ritanserin eye drops for 2 weeks. And I have to say, it doesn't look like there's a massive difference. It hasn't been as effective as the drinking water was. But we do all sorts of analyses on these things, and we showed that it's not doing any harm, that cell division is normal, that the gross health of the eye seems to be improved a bit, but not massively. And we can look at gene expression in these things. And this is the last science slide here. It's kind of backwards. We're looking at the thing on the right first. In this graph here, the higher the bar, the better. We're looking at expression of one of the proteins that holds the cornea together. It's called cytokeratin. It's there. It's one of the proteins that stops the cornea from falling apart. And normally in people with aniridia, the cornea doesn't produce as much of this protein. It's one of the reasons why it's fragile. And this is a healthy mouse eye, and it's producing loads of the stuff, big bar. And the control aniridic eye still produces some keratin 12, but not as much as it should. But after eye drop therapy, it goes right up back to normal. So we're restoring, using these eye drops, we're restoring normal levels of expression of one of these key proteins that stops the corneal surface from breaking up. The left-hand side of this science slide is something that I haven't really talked about yet, but this is using antioxidants. Given the aniridic mice antioxidants in their eye drops, does it reduce the stress? Does it sort of put out the fires that the aniridic cornea is normally fighting? And the answer is to some extent, yes. So in this graph, the lower, the better. And because this is a aniridic cornea with lots and lots of stress, a wild type, a normal cornea would have virtually none. It would be down at the bottom somewhere. And an aniridic eye that we've given antioxidant drugs to, the stress is reduced and the cornea is calming down, and hopefully that will give it time to heal. So this is where we are at the moment. We still have quite a lot of work to do. But I'm gonna stop, and I'm gonna summarize essentially where we are. So on this slide we have our conclusions on the right, and on the left is a piece of artwork we did which describes the process of working on aniridia, and it's some sort of science-based Sisyphus pushing a giant transgenic eyeball up a big hill, and every time he thinks he's made some progress, he wakes up next morning and finds that the eyeball is back down at the bottom of the hill. And he's in a landscape of inflamed eyes. But the essence of what I've been talking about is that we are trying to develop eye drop therapy that can be given to people with aniridia to either prevent the corneal opacity from ever happening, or in people where it's already happened, to stop it or to reverse it. And in order to do that, we are testing, we're using drugs that are already approved for something else, and asking the question: can they increase PAK6 dosage? Can they improve the health of the aniridic eyes? And we've shown that two of the candidate drugs that are already in the public domain, duloxetine and ritanserin, are really quite effective at improving corneal phenotype, the eyes of mice. And we are testing new versions of the drug ataluren. So again, many of you will remember there was a drug called ataluren, which has been used in clinical trial for aniridia. Hasn't really been very effective, and we're looking at a whole panel of things that might be better versions of ataluren. We're actually quite excited to find that antioxidants can be quite effective at reducing the stress within the aniridic corneas, because these are very widely available, and they're not even drugs. You can very easily dose up antioxidants, and it may well improve the phenotype, the symptoms of the cornea over time. And so, there's lots of things to be optimistic about. But as always with these things, there are lots of caveats, lots of questions, and lots of work still to do before we're at a stage where we're seriously proposing putting these drugs in aniridic eyes. I have to thank some people. So on this slide, this sort of grey pancake here is an aerial view of Aberdeen. It's not the most inspiring looking place in the world, although it's nice and sunny today, I have to say. We've been funded by Medical Research Scotland and the NHS Grampian Endowment Research Fund to do this work, so we are getting some clinical backing for this work, although as always we're short of cash. And quite a lot of the work that's described today wasn't done by me because I just sit around drinking tea. It's done by the postdoctoral person here. This is my lab. I tried to take a photo, but it went as well as you might expect. But most of the work has been done by this person here, Kaja Kastanjevets, with Jakob Yildirim, Teresa Senfeld, Salem Moreira is not on this slide, Anouk and Yvonne Brown. And of course, we're all based at the University of Aberdeen. So that I'm going to shut up. If you have any questions about the work, I'll be very, very happy to take them.
Thank you very much, Martin. That was really, really interesting work. So we do have some questions in the chat. The first one is, can you give some insight into the efficacy of the existing eye drops that many of aniridics already take?
I'm not really an expert in this, but I mean, eye drops that people with aniridia take will be primarily aimed at protecting the surface of the cornea. So prevention of dry eye is really important, making sure that the corneal surface is moist. Obviously, people may be taking anti-glaucoma drugs as eye drops, and that's very effective. There's no problem there, but there really remains no treatment that prevents the underlying causes of why aniridia-associated keratopathy happens in the first place. So, generally looking after the eyes is obviously a good thing, but there's nothing really. There's no therapy currently that, for example, increases the PAK6 dosage in the corneas. Everything is aimed at the symptoms, and there's nothing really associated with the causes.
Yeah, I think there's quite a lot of issues at the moment in terms of having that preventative treatment over dealing with the issue once it's already there.
Yeah.
We got a question with regards to what are the levels of the sort of dosage regarding the drugs that you're using compared to the dosage they normally are related to for their antidepressant and antipsychotic roles.
The good news is that it's much smaller. They are much more effective at increasing PAK6 dosage than the dosage that they are given as antidepressants. If you scale it down if you scale down the human dosage to what a depressed mouse might, for example, get, then it's the dosage we've been given to improve the eye phenotype is about a quarter of that. That still does have a subclinical effect, though, and although it's probably safe to give kids duloxetine at a very low dose, one of the real reasons we're developing eye drops is to avoid that systemic dose. If you give people the drugs as eye drops, it goes straight to the target, and the systemic dose that they get across the whole body will be essentially undetectable. So the summary is that it's a very low dose, but even so, we're trying to minimize it as much as possible, such that there's little risk of non-target effects. Because clearly, if we develop some eye drops, then the idea is that people will be taking them for their entire life. So that we have to be really careful that there's no side effects.
Yeah, that makes sense. Thank you so much. So next question, they said, like, you've done some amazing work over the last 20 years to get to this point. How long would you guess before it would be potentially available to patients? How much more work is involved?
That's always the million-dollar question, isn't it? So we work closely with some patients, and they ask the same thing. I wouldn't be doing it if I didn't think we could get something within the next 2 or 3 years. That's the target. Maybe even less. I mean, I've got a PhD student starting in October whose job is to start to take these things to clinical trial. So we're optimistic that we can do things quickly. But I'm very old now. I've had lots of disappointments in my life, and I don't want to give people false hope that there's something just around the corner. Unexpected things may crop up. There may be a delay. But so far, the signs are very good, and we're very hopeful that we can get something going within a couple of years.
Brilliant. I'm sure we all have our fingers crossed that your work goes smoothly. But yes, there's always trials and tribulations to work through. One of the last questions we've got here is, do you hope that this approach might be able to actually reverse current keratopathy damage that's already occurred in the future?
We're really hopeful that it can. One of the things that not just us, but lots of scientists have shown in the last 20 years is that the eye and the cornea has tremendous powers of regeneration. And that if you give it a chance, it will repair. And the problem at the moment for people with aniridia is as the opacity develops, the cornea is being overwhelmed beyond its regenerative capacity. If we can increase that give it a chance to regenerate by reducing the stress or increasing its PAC6 dosage, there's every reason to hope that existing damage can be reversed, that we can make the blood vessels go back and give the corneal structure time to repair. I don't see any reason why that can't happen. As with all human conditions, I think it's going to be better to catch it early and stop it from happening than to try and reverse it once damage is done. But I'm very optimistic. I believe in the power of eyes to regenerate. It's been shown lots and lots of times that the cornea is a tremendously adaptable and strong tissue, and I see no reason why we can't restore vision in people that have already started to lose it.
Brilliant. Thank you so much, Martin. I'm sure we're all very intrigued and can't wait to see what happens to your research over the next few years. I think we've got one final question here that's just popped up. So, in regards to antioxidants, are we referring to over-the-counter and contributing of good diet?
I'm very tempted to say that eating and the good diet can't be a bad thing, because good diet is never a bad thing, I guess. What we've been looking at is so there are some over-the-counter antioxidants, that eye drops that people get for various inflammatory conditions, and we're not using that. We're using some of that. We've been using some of the components of them. The reason we're not using over-the-counter medications is that they've been reported to be quite irritating for the eye. That they sting a bit when they go in, and that's try and persuade kids to do that one. But also because the aniridic cornea is fragile, we don't want to cause anything that's going to be an extra irritant to it. So we're looking at over-the-counter antioxidants, but we're trying to see what we can do with them that makes them safe for aniridic eyes, because at the moment I'm not totally sure they're a fantastic idea for people with aniridia.
Brilliant. Thank you so much, Martin.